Good afternoon, my name's Dan Macqueen. I'm based at the Roslin Institute in Edinburgh and I'm the Deputy Coordinator of AQUA-FAANG. In this short talk, I'll introduce the project and give a quick overview of its progress before handing over to my Roslin Institute colleague, Ross Houston, who's leading a work package on the breeding associated goals in the project and will provide an overview of that effort. As a brief rationale for the project, you'll be aware of the importance of aquaculture in global food and economic security. And aquaculture is a rapidly growing industry involving a much more genetically diverse group of organisms compared to terrestrial farmed animals. Aquaculture species also have markedly shorter domestication timescales than terrestrial livestock, and consequently, there's more extensive scope for rapid genetic gain and trait improvement in modern aquaculture breeding programs. Infectious diseases represent a major challenge across all aquaculture sectors and species. And this is a big hurdle we have to overcome if we're to ensure a sustainable expansion of this important global industry. So genomics already plays a big role in supporting genetic management and enhancement in aquaculture. And this was illustrated by a Nature Reviews Genetics article that was led from the Roslin Institute recently that I've shown a figure from here. For example, most of the main aquaculture species have high-quality reference genomes these days that are widely utilized in research orientated towards breeding and genetic management goals. Functional annotation, as outlined in Elisabetta's opening talk, is very important to being able to understand and ultimately exploit the functional genetic basis for commercial traits. This sort of work is even more limited in aquaculture species than in livestock. AQUA-FAANG then addresses the gap in functional annotation and understanding of the functional genetic basis for commercial traits, focusing on the 6 most commercially important finfish species in European aquaculture. How are we doing this? We're doing this by generating novel maps of functional elements in the genomes of these species using several of the technology approaches outlined in Elisabetta's talk. And this new information will be used to identify genomic regions involved in commercial trait variation, in addition to prioritizing and exploiting the potential of causative genetic variants in breeding, all with a major focus then on improving disease resistance. The overall concept of AQUA-FAANG is to make a major step forward in understanding of elements in fish genomes responsible for the regulation of genome activity and ultimately of gene expression under different biological conditions, for example, including both healthy and immune or disease-challenged fish. In other words, we want to make a major improvement in genome functional annotation, targeted at gaining knowledge surrounding disease resistance mechanisms. And to do this, we have 5 project-specific objectives, and without going into details, the most important objectives to highlight in this talk are using the new functional annotations to be able to predict disease resistance, to ensure the results are actually converted into practice for sustainability and economic benefits in aquaculture by ensuring they're engaged and shared and used by stakeholders. More specifically, the project's been developing new approaches to identify regulatory elements that are controlling gene expression, including ATAC and ChIP-seq, with several first applications of these tools in the 6 target species, alongside more established RNA sequencing-based methods to identify gene expression phenotypes. This is being done across a range of different life stages and in both healthy and immune-stimulated tissues and cells. And the data that will be generated will be used alongside additional research being performed in the work package that Ross is leading on, with the aim to exploit functional annotation information and understand its applicability or added value in genomic-orientated breeding applications like genomic selection. There's too much progress to highlight in the short slot that's available, so just a few headlines before handing over to Ross to talk more on the breeding-focused goals of the project. So we've developed robust functional annotation protocols for these tools to identify regulatory elements, both the ATAC-seq and the ChIP-seq. The project partners have also delivered greatly improved reference genomes for 4 of the 6 targeted species in the project. Those genomes and all the functional annotation data generated in the project will be made available through the Ensemble genome browser. Functional annotation data has been generated using these methods mentioned across all the 6 species in a large number of samples. That includes embryonic developmental landmarks, which we call DEV maps, a tissue panel from both juvenile and sexually mature fishes of both sexes, which we call body maps, and immune tissues and cells subsequent to viral and bacterial stimulation, which we call immunomaps. And in total, we've sequenced close to 2,500 sequencing libraries to date. This data is now being analyzed currently and will hopefully all be generated by the end of this year. In addition to that, within the project, we've been developing approaches to better interrogate disease resistance phenotypes using single-cell RNA sequencing, which is a novel technology in fishes, and also by using in vitro CRISPR-Cas9 gene editing and developing tools in this area for actually the ultimate level of functional validation of elements and variants in fish genomes.
Okay, thanks very much, Dan. I'm going to talk a bit about Work Package 5 of AQUA-FAANG, which is really where we're trying to harness some of the functional annotation data coming out of the other work packages towards improvements in selective breeding, particularly focused on disease resistance. But a bit of background first. So genomic selection is really now becoming state of the art in several aquaculture species, particularly in Europe. And just to illustrate for this audience generally how it's done within a finfish breeding program, we utilize the large full sibling family sizes that are possible with fish to create large reference populations with many representatives of each family. It could be perhaps 100, 200 families within a breeding program. That reference population will receive, in the case of disease resistance, a disease challenge from which disease phenotypes and genotypes will be collected. And those data are used to train the genomic prediction model in a similar manner to how it's done in terrestrial livestock, such that selection candidates are chosen on the basis of breeding values calculated using genotypes only. And it's typically performed with genome-wide anonymous markers using SNP arrays. How well is genomic prediction performing in aquaculture? Well, we did a review study that Dan alluded to earlier last year, where we looked at that stage 36 studies across 18 species that had a high diversity of traits that were studied, SNP densities, sample sizes, different genotyping platforms. But the message was rather consistent that there was a substantial increase in accuracy using genomics versus the pedigree. For growth-related traits, the increase was on average 24%, and for disease resistance-related traits, on average 22%. And this is largely thought to be derived from the improved ability to distinguish between siblings in the large families that I alluded to earlier. So that's a good news story, but the challenge is that accuracy rapidly drops with genetic distance between the reference and validation populations, and you can see that starting to happen in the figure on the right. And once you lose full and half siblings from relationships between these sets, the accuracy actually drops markedly and can be close to zero. So that's one of the challenges we're looking to address in Work Package 5 of the AQUA-FAANG project, asking the question of, can functional annotation data improve genomic prediction accuracy and its persistence across more distant relatives? To do that, we have a large-scale exemplar study into resistance to viral nervous necrosis, which is a really problematic viral disease of European sea bass in Mediterranean aquaculture. This work has been led by Luca, and the analyses done so far by Robert there. The experimental design is represented here. So we have the 25 male and 25 female parents. All of those have been whole genome sequenced at a coverage of 15x approximately. There's approximately 1,300 offspring, the majority of which were challenged with VNN and sampled for survival or mortality as the key trait, with viral load measurements also taken. A minority were euthanized early and sampled for RNA sequencing in key tissues to look at gene expression alongside a smaller population of controls. The idea behind that is that the genomic breeding values of those RNA sequenced animals can be used to distinguish gene expression response between resistant and susceptible fish post hoc. So all the offspring were genotyped with a 35K SNP array, generated via the MEDAID and Perform Fish Horizon 2020 projects, and are being imputed to whole genome sequence. So we'll have a population scale whole genome sequence data set. So far, the heritability estimates look good. So with pedigree estimates, it's around 0.37, and with genomic, it's around 0.34. So this is very much a work in progress, and one of the next steps is to perform a genome-wide association study using the imputed whole genome sequence data to identify QTL affecting resistance to VNN. We'll then use the sequence data alongside the gene expression data and expression QTL identified within this work package, and join up with the functional annotation data coming out of the previous work packages to perform a detailed functional annotation of these QTL regions and identify putative functional variants, which we then plan to test in genomic prediction models with differential weighting on those variants. We're in discussions with BovReg and GeneSwitch colleagues to ensure we're learning from each other and applying best practice. And then we hope to develop functionally enriched marker panels for application in breeding and testing the application of these methods and tools across other target aquaculture species. So just finally, a bit about how we're going to translate some of these results to practice. Well, there's a few mechanisms, one of which is we have within the project an exploitation working group, which is chaired by EFAB. And that's very much a platform for cooperation and collaboration with our stakeholders, which include aquaculture breeding companies. And that's ongoing throughout the project. There's also a series of online learning and training events, again led by EFAB, which will facilitate exploitation of functional annotation data by these companies later in the project. And we're planning to produce a white paper, which is led by aquaculture genetics companies Elect in collaboration with other industry partners about how functional annotation data can be used to advance European aquaculture.